Welcome everybody to this lesson of the course, and I'm very glad to give these lessons that will be focused on a particular family of biomarkers of early effect, which has a number of implications, as you will see during the lecture. My name is Stefano Bonassi, and I'm a full professor in hygiene and a specialist in molecular epidemiology, in particular, exactly on the use of DNA biomarkers of early effect, especially of DNA damage and genomic instability. The lesson of today will be focused on the biomarkers of effect. And actually, the title of the lessons is A Journey from Exposure Monitoring to Predictors of Adverse Health Outcome. And in these lessons, I really wanted to go through the use of this biomarker, which started actually for monitoring the exposure to something more. The idea, we will see that a damage in the middle of the pathways to the disease, the pathway linking the exposure to the disease is something that may be really important for measuring and preventing the effect of the disease, but also of the exposure, but also to predict the risk of disease. So offers a big tool of prevention, since it can anticipate the diagnosis of disease, or may allow to identify the group which has a higher risk. When we talk about biomarker of early events, that is a very historical, iconic picture, in which it was very simple, in which the idea that the link in between exposure to the disease, while in the traditional epidemiology is a black box, is now, that time, that was the late 80s, was fragmented in different steps. Each of these steps, which was modified, possibly the effect of each damage in this step could be modified by the presence of individual susceptibility. So there was the idea that an internal dose, biological effective dose, early biological effect. Since the very first model, the presence of these biomarkers was considered, and then there would be late stages and diseases. The same idea was extremely developed, and so you can see a much modern model in which was developed, published a couple of years ago in a paper that was the result of a study group by OECD. OECD, in a way, is the international cooperative agency, which provides a number of rules that are included in national regulation and for preventing exposure to, especially in occupational workplaces, at workplaces, to prevent damages from exposure. And you see that actually there is a number of sub-classification from exposure biomarkers that can be measured in the blood, in the urine, and biological effective dose, which is the idea of a dose that actually perhaps go through enzyme activation or whatever, and then can create an effect. And the effect, the marker of effect, includes very early events, but also structural changes along the pathway. And you see that there is a number of these events can be classified very simply, like in these arrows, bringing to the health effect or to the diseases, but also other classification can be used, like early key events or late key events, or which are this biomarker are part of what is called adverse outcome pathways. The idea is that we can place these damages in a pathway. So these pathways put together mechanism put together epidemiological evidence, biological evidence, biomonitoring, and study to anticipate the diagnosis. So I just gave you a very brief overview of what is classified, what is the general consideration of those steps linking exposure to the effect. You see that it's quite complicated, but obviously once you put into play also genes and proteins and this scheme what can become really complex. Now this is a very oversimplification, but is useful for toxicologists and for preventive medicine people, professional of preventive medicine. Now let's go a little more into the detail of biomarkers of effect. So biomarkers of effect sometimes are called also biomarkers of biological response. Can be observed and they measure biological changes that can be quantified and the result from exposure to, especially to contaminants, chemical contaminants, but also to ionizing radiation, UV light, and so other physical or other kind of exposure. These changes can occur at different levels. I mean, levels of molecules, at cellular level, at organism level, at tissue level, and however they represent a number of changes that are due to exposure and may anticipate the disease. This is the strength of these biomarkers. And we are familiar with these biomarkers because you see that there is a number of biomarkers of effect that are of use in clinical medicine, clinics. Think about cholesterol. Cholesterol that can anticipate the risk of myocardial infarction or major cardiac diseases. And so we measure the level of cholesterol, which is associated maybe to a bad diet or a bad genes or low physical activity or an unfavorable genetic profile and predict cancer and cardiovascular diseases. So maybe it's associated also to cancer. So it's in the middle, and in the middle is a big power because can help with reducing exposure and help with preventing the disease. There are many others like bilirubin, creatinine, liver enzymes. And uric acid and so on. And all of these are linked to or a genetic unfavorable condition or an environmental exposure or an endogenous exposure and are specifically associated to a disease. So can be used for the prevention of that. And but when we talk about toxicology, in a way, we are especially interested in a kind of biomarker which is linked to the most common exposure. And then, however, you can see that DNA damage and the genomic instability as measured by the COMET assay or the micronuclei, but also the presence of sister chromatide changes. When we talk about oxidative damage, so the level of 8-hydroxydeoxyribonucleic, the formation of gamma H2AX foci, the mutant prevalence frequency of HPRT genes, obviously telomeres length shortening, and there is a number of, but there are many other like DNA adducts, for instance, or epigenetic changes. I mean, all of that, I linked just a few of them because there are several. And however, if you look into the PubMed and you see the biomarkers of this family, which are most often cited in the publication, you see that the COMET assay, the micronuclei are for sure, maybe also telomeres, but telomeres may have also other purpose. like a marker of aging, accelerated aging, which is a feature of many diseases, but not necessarily able to prevent the disease. In many cases, it can be a marker of the disease. And however, since I told you that this biomarker of like comet assay measured with the comet assay or the micronuclear assay are very popular, in a way I will focus on these two markers. And there is a kind of analysis which is called the SWOT analysis, which is commonly done in business administration, because to evaluate the strength and the limit of, for instance, a service, a department, an enterprise. And so in a way, in this 2x2 table, in this 4-square table, there are listed the strength, the weaknesses, but also the opportunities and the threats. That what are the threats to this biomarker that can stop its usefulness or may change the consideration about this. So when we talk about biomarker of DNA damage and genomic instability, you have the high sensitivity to DNA damage is an advantage. It's not invasive. You can use buccal cells. It's suitable for various cell types, cost effective for various cell types. It's simple and a number of can be tailored for specific damages. So as a valuable biomarkers for many purposes. There are also some weaknesses, like the lack of specificities, the limited information, long-term effect of these damages. So we observe the damage, we are not sure what happens in people with a lot of this damage. And there is other measuring, for instance, can be limited the reliability of scoring because of heterogeneity of scoring. I mean, there is in the balance some strengths, some weaknesses, but there is also some opportunities for the future, which is but not for the future, even for today, because the simplicity of these biomarkers and make this biomarker, especially comet assay, useful for large population sampling, for large environmental studies in large populations. And another important issue is dealing with personalized medicine. I mean, personalized medicine, which is a very, very popular approach nowadays, and kind of approach that intends to go over the problem of evidence-based medicine. So there are opportunities, but there are also possible threats. The most important is that people may stop trusting this biomarker, which is in a way are quite simple, preferring high throughput approaches like genomics, transcriptomics, proteomics, metabolomics, I mean all the omics. So in a way, the idea, obviously, these biomarkers of high throughput and coming from high throughput analysis are definitely important and maybe really predictive and very specific. But over specificity in some cases may be a problem. And much better in some cases to use a sort of comprehensive biomarkers like this one. And some problems with this biomarker were the problem, the heterogeneity of the protocols. So different laboratories use different protocols, the scoring. And so there was the fault of, this is the target of the efforts of collaborative international projects. And for instance, the H-Comet project has published a couple of papers on the natural protocol just to identify a common protocol. And so the use of a common protocol can reduce greatly the variability of the assay, which becomes much more reliable. And the idea of using biomarkers of effect which are reliable, which are standardized among different laboratories, provides the possibility of provide the possibility of using this biomarker actually for the safety in occupational health. And this is the interest coming from OECD, some other countries is called OCSE. You may be more familiar with the French, Italian, Spanish name for this agency. And of actually using this biomarker to integrate this biomarker in risk assessment, and especially in the individual risk assessment. That, by the way, can be used also in regulation for improving and actually taking place the occupational safety. The point is that the first work of this effort was just to understand what's the role, how are used today biomarkers of early effect. And that's actually, they are highly underused. And there are kind of biomarkers which are very good for mixture. Mixture is the major problem that we are facing nowadays. And these kind of biomarkers are the only option that, not the only, but is the most reliable option in which you can measure the effect of different exposure and summarize the damage and summarize also the potential for prevention. And once we have validated these biomarkers, can be used, to understand also the adverse outcome pathways and the mechanism of action of this damage, of this exposure, linking the exposure to a specific diseases. And okay, there is something more that you can read, and this biomarker obviously can be used on the human biomonitoring to protect the humans, especially in the workplace, but also into the environmental monitoring to protect humans living in near a polluted area, but also animal species and different species that can be used or as a marker, but also as a target of environmental pollution. So the point is that we need really to identify which biomarker can be measured, which reflect which damage, and what is the intensity of response that we have to measure that can imply a damage long term. Then we have to shift back to the biomarkers and to the literature, because link between these biomarkers and disease is a dream for people working, establishing a link between the frequency of this biomarker in healthy people and the risk of cancer, of other diseases in the long term, is really which is a validation of this biomarker as a predictor, is really important, because it can realize the success of this biomarker and to increase a lot the use of these biomarkers especially for prevention, not only for exposure monitoring, but also for preventing, for individual risk assessment. You see, for instance, now we are moving to another biomarker, which is the micronuclei. Micronuclei frequency in lymphocytes is measured after dividing cells, adding cytokalasin that block the division of the cytoplasm. So we have a binucleated cells, which is very easy to measure the presence of micronuclei, which are this small formation resulting from the cell division. And micronuclei is very commonly used to measure exposure, but also has been commonly linked to cancers or to other diseases. You see that here is the mean ratio, which means that is the ratio of the frequency of micronuclei in people with a disease to the people without a disease. So, for instance, diabetes is like 4 times higher frequency of micronuclei,
or child diseases or autoimmune diseases 3 times more. And the same for cancer. You see that especially blood cancer have like 5 times, which is not surprisingly, since this damage is measured in lymphocytes, but also solid tumors have a much significantly higher frequency of micronuclei in people with the disease. You know, the problem of this approach is that uh, there is a problem of reverse causality. So when we measure with a cross-sectional approach cancer and genetic damage may be induced by cancer. So you don't know it actually if it's unlikely, but you don't know really if the damage is induced by the cancer or is the damage that induced the cancer. Then validation must go through prospective studies. And cohort studies are just those studies which are very robust to bias. And why? Because we start from people which are, all of them are healthy. So you measure micronuclei, these people, or DNA damage or chromosomal aberration or other. This same study has been done in several different biomarkers. You measure this damage in people which are healthy, and then you follow them up for 10 to 20 years. This can be done with a specific study design, which is called historical cohort approach study. And then you can measure, and you see that people with a low level of micronuclei in the lowest tertile has a much better, significantly better survival than people in the high and the medium tertile of micronuclei frequency. So especially for macronuclei, that's the idea of the OECD. That from the literature, there is enough evidence linking high level of macronuclei to a higher risk of disease. So for this purpose, there is an ongoing project to establish thresholds, establish rules that can be applied in national regulation and to protect more and more the health of people at workplace. So the idea is to provide a baseline micronucleic frequency, which is specific for each lab to take into account age and sex, so a baseline reference. So once we have a baseline reference for each laboratory, so we can take into account age and sex, which are very important predictor of micronucleic frequency. So once we have a baseline level, we can measure micronuclei in people at risk, people that are exposed to genotoxic compounds. Then we can measure the ratio. And there is one further step, which is say, okay, the study was done well. So the study is reliable. So the idea of reporting the strobe molecular AP rules, there is a checklist listing some issues that must be reported to establish that the study was done properly. And then once this approach has been done, the idea of using this traffic light approach, in which if you don't have any increase in people exposed or working in a place where there is genotoxic compounds, nothing should be done. If there is a significant increase but is low, below a doubling of micronuclear frequency, I mean, we know that there is some kind of a genetic exposure, but yet no strong regulation should be applied. While when there is a higher increases due to exposure, and this increase is significant, is more as a double or more than double, then regulation can apply. So workers must be moved, new the protective devices can be used, the use of protective devices should be implemented, and so on. So this is quite that's very nice for people working micronuclei, the idea that you can translate from the lab to the actually to the security of workplaces. What next? Next is made stronger and stronger the association between biomarkers and the disease. And this imply also the possible use of the biomarker in clinics. That if you think, if you look into the history of cholesterol, in the past, cholesterol was not very different from chromosomal aberration or micronuclei or DNA damage, in which there was the evidence that there was increase, but nobody really applied to clinics until there was cohort studies and randomized clinical studies, randomized controlled trials, sorry. And actually, this is exactly what is the more important steps in the process of validation of a early effect biomarkers, in which once you have data from a cohort study and randomized controlled trial, then the link with the disease is well established and is quantitative. So you can really apply for individual risk assessment for disease prevention. Obviously, there is a final step which is linked to pragmatic trials, but especially to the trust of professionals in the field. And as I told you, this biomarker can be extensively used, have many potential use in the clinics and clinical trial as well. So, for instance, they can surrogate endpoints, can allow early detection of drug response, and especially can be used for personalized medicine. For the purposes of toxicology, actually the idea is that the use of this biomarker allows the prevention and risk reduction and the early detection of diseases, so making stronger the possibility of prevention. And I'd like to close my lessons with a few slides just talking about something different, but I believe that when you work as a new toxicologist, you work a new toxicologist that take into consideration the complexity of the interplay in between its different compounds, different exposures, but also different sensitivity of the people and the early events and the exposure, the biomarker of exposure, the genotypes that identify protective of those polymorphic enzymes that can accelerate or protect from exposure. In these compounds of different agents that can modify the effect of exposure are also viruses. And viruses are important because there are a number of studies that linked the frequency of, for instance, the coronavirus, that we know exactly what we are dealing with some kind of exposure. For instance, nicotine. Smoking, the effect of smoking can interact with COVID-19 in the etiology of COVID diseases. But also there is a number of viruses which are present in many humans. That for one of these are torquetanus virus, which is the same family of the coronaviruses, is extremely present worldwide. Maybe 80–90% of people is infected. What makes the difference is the extent of the infection, which is the extent of the number of copies of the virus which is part of infection. And so that may be really modifying the effect of exposure. And I wanted just to tell you that we have investigated some group using DNA damage marker and measured with the Comet assay. And what was clear was that look at the interaction between the DNA damaging effect of TTV, torquetanus, in which you see that people with four copies or less of TTV has much lower DNA damage than people with 4 or more or 6 copies or more. This is quite, there are significant results and a nice trend. These are patients with COPD, but also patients without diseases can, or with other diseases can demonstrate the presence of an increased DNA damage leading to genomic instability that in turn can accelerate aging or the progression of the diseases, especially if there are cancer. What is important to know is that, for instance, just to tell you how important can be the TTV infections. For instance, this is we did this study in people with COPD, and we measured the presence of the number of copies of TTV, and you see that that was quite clear that people with uh, uh, less copies of TTV with a lower infection, the less important infection is a much better survival. These are a mortality study on a group of little more than 100 people with COPD. So they were quite advanced, old people. So we had a good number of people dying. So the numbers are reliable, and you see that people with the higher level of infection has much lower probability of surviving, and that was also interesting. If you look at survival and DNA damage again, you see that the people, it's the same condition, the same population as before. But if you see that you separate people with a high level of DNA damage, which are the red line, and low level of DNA damage, which is the blue line, you see that there is a strong univariate difference in between the survival probability of these patients. But what was interesting that when we try to move from univariate analysis to multivariate analysis, you see that the 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 TTV that was quite clearly showing an effect of mortality. So it was a good predictor of mortality. When we adjusted by DNA damage, this this evident difference in survival disappeared after adjusting for DNA damage. And in a way, that was quite interesting because demonstrated that, or not demonstrated, maybe too strong, but supported the idea that the effect of DNA damage due the effect of mortality associated to viral infection can be go through DNA damage, because once you adjust for DNA damage, you don't see any longer effect of TTV. So this was a very highly specific example, but I wanted just to encourage you to the use of biomarker of effect, and because they are really predictive of diseases, many diseases. And this may be a problem because there is low specificity, but there is high predictivity of several diseases, which in a way is good for prevention, maybe still associated to exposure. And what is important is really that to have the more complete, comprehensive information and ability to model the predictivity of this biomarker is to take into consideration as many confounding or potential confounding effect modifiers. I gave you the example of viruses, but also to take into consideration smoking, diet, physical exercise can be something that can be really modified, the predictivity of these biomarkers. That's it. Thank you for listening.